Well, I'm really excited to introduce Lori O'Brien to everybody. Lori is going to talk to us today about new models and methods for interrogating kidney development and disease. And if you get the chance to, follow her work a little bit more closely after this talk, I think you'll really enjoy some of the things that she's doing. These beautiful images that are on the side of the screen are hers. And so there's just some really fun science and some really pretty science that's happening. Lori got her PhD at University of Wisconsin in Madison. And then she did her first postdoc at Harvard with Alan Davidson. And it was a short postdoc because he then moved to New Zealand. But in that time, she did some really great work on podocyte development in zebrafish and how Notch and WT1 and FOXC1/2 can regulate the number of podocytes, which, as I'm sure she'll tell you, is really important for kidney function. Then she moved over to the lab of Andy McMahon, also at Harvard, until he moved out to California, and she followed him there. Since that time, so he's also a big kidney development person. And so since that time, Lori has come to the University of North Carolina at Chapel Hill, started her own lab, and she told me that she had to get back to the podocyte because she just loved them, and I relate to that so much. They're really a great cell, a really interesting biological question as well. So without further ado, Lori, take it away.
Great. Thanks, Mariya, for that very kind introduction, and thanks everybody for the invitation and the opportunity to speak and for attending today to hear about some of the work we've been doing. So today I will be debuting a few new stories within the lab, and both stories, so they're a bit different from each other, but both involving new models that we're trying to develop to understand kidney development and disease. And so this will be split kind of between springing off some of the work in Andy's lab and looking at nephron progenitor cells and how they lead to the development of Wilms tumor. And then secondly, then the other part of my talk, I'll be talking about podocytes and a new model we're trying to develop to understand changes within podocytes, specifically in the foot processes at the slit diaphragm that are occurring with disease to hopefully come up with some new therapeutic targets. So so this is all new, so hopefully this goes relatively smoothly, but if you have any questions, at any time, anybody can feel free to interrupt me. I don't mind if you need clarification. But with that, whenever I do this pointer thing, the first slide is like always impossible to get past, but all right, here we go. This should work now. All right, so just basics of the kidney for those of you who aren't nephrologists or work on the kidney. If you know, the kidney is what's doing all the blood filtration, and the blood filtration occurs, so it's really the nephron, which is the filtering unit of the kidney. So, we think of the kidney as a blood filter, but it's really these nephrons which are doing this really important function of blood filtration, where you have the blood going in here to this glomerular structure, which I'll talk a little more about later. And then this filtrate from the blood goes through this tubule where it gets modified and eventually makes its way into this collecting duct system where it will eventually be excreted as urine. So as I mentioned, blood filtration and also, the output of this is really to maintain body fluid homeostasis. And so in the adult human kidney, there's about 1 million nephrons that are all functioning together to work to filter your blood. So now stepping back to kidney development and how you make these nephrons in the first place, in this schematic here, I'm showing you what happens in the mouse, and most of what we know about kidney development we know from the mouse, but I'll touch a bit on human kidney development as well. So if you see embryonic days, those are the embryonic days in mouse, and then the relative weeks in human gestation next to it. So kidney development really kicks off with this invasion of this nephric duct, this epithelial tube, into the adjacent mesenchyme. And this sets off around, multiple rounds of branching, which is going to help develop this collecting duct system. And additionally, these progenitor populations coalesce around the tips of this branching collecting duct system and really serve as the progenitor pools for all the cells within the developing kidney. So in this, we have this initial branching happening here at about E11.5 or 5 weeks in the human, and the nephron progenitor cells are going to cap around this ureteric bud tips and be maintained around those tips as the branching program continues throughout development. And at the same time, those nephron progenitor cells are also going to start differentiating and making the nephrons of the kidney. And this continues through about postnatal day 2 in the mouse and about 36 weeks of gestation in the human, at which point in time, all those nephron progenitors undergo their final wave of differentiation and you've formed your 1 million nephrons. So as you can imagine, kidney development requires this careful balance of self-renewal to maintain this progenitor pool for a sufficient amount of time, but at the same time also differentiate those progenitors and form a sufficient number of nephrons. So going from this very, early branch structure to this, quite arborized structure with all these progenitor pools, we've obviously multiplied this population of progenitors, but at the same time, there's all inside of this kidney here, there's nephrons that have formed as well. But we know if we, have any imbalance here and insufficient nephron progenitor pools, this can actually lead to a nephron insufficiency because you don't have the progenitors there, a sufficient number to form all the nephrons within the kidney. And this can predispose these patients to hypertension or kidney disease. And as I mentioned, by the time you're born, you've formed all your nephrons for life. There are no adult nephron progenitor cells. So, again, this requires this very careful balance. However, there's also sort of the counter to this that, potentially, you can have, proliferation of this progenitor pool, and this will actually lead to the development of this pediatric kidney cancer called Wilms tumor. The Wilms tumor is the most common childhood kidney cancer and is typically diagnosed around age 2 to 3. And it can present with multiple different histologies. And interestingly, these histological features of these tumors look a lot like the different cell types of the kidney, either representing this blastemal cell type, which actually kind of look like those nephron progenitor pools that are present within the developing kidney, epithelial structures, which are like the nephron tubules, or the stromal cells, which are all the other cells surrounding these nephron structures within the kidney. And additionally, you can have this mixed histology as well. The blastemal tissue, interestingly, actually resembles nephron progenitor cells. And so, like I said, they resemble them in terms of their morphology, but also in terms of their gene expression. And it's the prevailing hypothesis is that they actually develop from a malignant transformation of those nephron progenitor cells during development. And why we're interested specifically in this blastemal type is that these blastemal predominant tumors are actually classified as high risk. They have higher rates of relapse and compared to the other cell subtypes of this tumor. And within the last several years, there's been groups interested in, what are the mutations associated with Wilms tumor? And it turns out that after sequencing a number of these tumors, specifically the blastemal predominant tumors, where the cells look like the nephron progenitor cells, they identified recurring mutations. And I'm going to focus today on 6.1, this recurring mutation in this homeobox transcription factor. And so this is just an example of a tumor section here where they stain for 6.1, and you can see 6.1 being expressed within that tumor blastema. The mutations are somatic and they're also heterozygous, except in very rare cases where you might find a homozygous mutation. And so this really suggests that, this mutation is really driving, the transformation of these cells and driving the tumorigenic program because it's acting in sort of this dominant fashion. There's also a mutation in 6.2, related family member, another transcription factor, and I'll show you in a minute, acts similarly as 6-1, but it occurs less frequently. And there's equivalent expression of the mutant and wild type alleles. So this isn't a case of overexpression of this transcription factor causing this cancer. And this recurring mutation occurs in this residue 177, and it's a highly conserved DNA contacting residue. So as I mentioned, it's a transcription factor, and it has a DNA binding domain, and this mutation occurs within that DNA binding domain, specifically at this point of DNA contact. And this point is conserved from a 6-1 to 6-2, and also from human to mouse. And so, you know, the question is, how does this then lead to this tumorigenesis, and why am I focusing on 6-1 here? And why is this important in terms of kidney development? There we go. So it turns out that, you know, work from several other groups, and including our group, we know that 6-1 and 6-2 are actually really important for kidney development. So 6-1 and 6-2, turns out if you look at knockouts of these animals, in the case of 6-1, you get complete renal agenesis and you do not form a kidney at all. And in case of 6-2, when you knock out 6-2, you get this very small, tiny kidney where you've only formed a few nephrons. And that's because 6-1 and 6-2 are expressed in these metanephric mesenchyme, that early mesenchyme, where that ureteric bud is going to come over and invade. And then 6-2 is remains on in those nephron progenitor cells. So there's an interesting dynamic between 6-1 and 6-2 in the mouse, with 6-1 being on very early and required to maintain this early progenitor pool. But it actually gets rapidly shut down, specifically in those nephron progenitor cells. This is just an adjacent non-kidney cell population here labeled by 6-1. And it is never again subsequently expressed within those nephron progenitor cells of the kidney. However, 6-2, it's expressed at the same time as 6-1 in that early mesenchyme, but it stays on within those nephron progenitor cells throughout the remainder of development, and is actually required to help maintain that nephron progenitor pool, because as you can see, when you get rid of it, you get these very small kidneys where you've now depleted that nephron progenitor pool. Interestingly, though, what we found in the human kidney is that 6-1 expression is actually maintained in those nephron progenitor cells, unlike the mouse, and it's maintained, and you can see it within the same population as 6-2. As I mentioned, in those progenitor cells that persist out to 36 weeks of gestation. And through some work back in my postdoc in Andy's lab, we wanted to know, do 6-1 and 6-2, you know, do they act the same similarly as transcription factors within this nephron progenitor population? Do they both help support the self-renewal of these progenitor cells? And the way we approached that was to do ChIP-seq from human fetal kidneys. And we actually did both 6-1 and 6-2 ChIP-seq from human fetal kidneys, and then compared them to see, you know, what are the binding sites the same between 6-1 and 6-2? Are they binding at the same points in the genome? And also, are they targeting the same genes within the nephron progenitor cells? And so this is just to show you, because they have that very highly conserved DNA binding domain, they are binding actually the same motif. So they have the same DNA binding preference between these two factors. And this motif was enriched to the same amount in all the binding sites that we looked at. And when we overlap the peaks being, you know, the sites where 6-1 or 6-2 are bound within the genome, we see that there's actually significant overlap. And similarly, when we look at the putative target genes of 6-1 or 6-2, we can see again that there's really significant overlap. And when we do gene ontology analysis of those putative target genes, we see that they are involved in kidney development. Specifically, you can see here terms such as metanephros development or nephron morphogenesis, and tissues also where they are enriched, such as the metanephric mesenchyme. So this really, to us, suggested that 6-1 and 6-2 are probably working together in this nephron progenitor cell population to help maintain them for that whole length of gestation and keep them around for that total of about 30 weeks. They need to be around and keep self-renewing. Oh, and this is just another thing, looking at these actual ChIP-seq peaks, just, I showed you the overlap in those little circles, but just if we go and now look at the genome, so look around 6.1 here, the 6.1 locus, we can see that 6.2 and 6.1 are both bound at the same enhancer for 6.1. And similarly, 6.1 and 6.2 are both bound at the same enhancer for 6.2. So there's, sort of this cross-regulation and auto-regulation really driving the expression of each of these genes within those nephron progenitor cells. So this comes back then to that mutation. So what's happening in the case of these nephron progenitor cells that acquire this Q177R mutation? And what is that doing to help promote, is it promoting the self-renewal of these progenitors so that they turn tumorigenic and can't differentiate? And so we wanted to utilize some publicly available Wilms tumor data to be able to start looking at this. So, and compare that to our human fetal kidney data. So, taking an approach where we don't really have to do many experiments for this, but really just take this publicly available data and our own data and start looking at how are the programs modulated by this Q177R tumor or mutation different than programs that are modulated by the normal 6.1 in the human fetal kidney. And so we took 6.1 ChIP-seq data. There was a group, Manfred Gressler's group, that actually found one homozygous tumor for this mutation and did a ChIP-seq from it. So we had one data set to go with where they actually did a ChIP-seq here. And now we could compare that to our human fetal kidney data. And also there was RNA-seq data, and we could take and integrate these two data sets to find genes that were differentially expressed between, a wild type 6.1 and the mutant 6.1 tumors and the human fetal kidney. And also they were putative targets of the mutation. So really suggesting they're directly regulated by that mutation, and also that's driving its increased expression within these tumors. And so as I mentioned, this data from Manfred Gessler's group, they had tumors that were wild type for 6.1, so no 6.1 mutation. And they had tumors that carried the 6.1, the one single tumor that carried that Q177R mutation. And they did ChIP-seq from both and looked at, are they sharing common binding sites? And it turns out there are. And if you take those shared sites and you overlap them with ChIP-seq from the human fetal kidney, which I just showed you, there's actually a significant overlap. So it seems that, where these transcription factors are binding in these tumors are driving programs very similar to the nephron, what 6.1 is doing within the nephron progenitor cell population. And so looking at the motif discovery, so as you might expect and predict that, if you have this mutation in this DNA binding domain of this transcription factor, you might expect that it actually binds to a different DNA sequence or a different motif. And so looking at this data, so this was going back to that motif that we pulled out from the human fetal kidney data for 6.1. Similarly, if you look at the wild type tumors for 6.1, the motif, as we would expect, is actually very similar. And those shared peaks I showed you, which are where both of these wild type and mutant proteins bind in the tumor, again, you find a very similar motif. But what was, striking is that when we looked at those mutant tumors, we now identified this differential motif, specifically this left-hand side of this motif here, where you had now this different preference of binding. So again, this may suggest that, this mutant protein is driving different programs that may help, promote the tumorigenesis of these cells. And just to confirm that, we did a collaboration with Martha Bullock at Harvard to confirm that, this was that, since it was just one tumor that we're looking at, one ChIP-seq, we wanted to confirm that, this was a preference for this mutant protein for that specific motif. And so they did these protein binding microarrays. And so what this is you just have all these oligonucleotides stacked up here that are essentially every sort of 8-mer combination that you could have. And they're arrayed down on this plate. And then you take your tagged protein here and you put it onto this plate, this array, and it's going to bind to any motif that it has an affinity for. And then using a fluorescently labeled anti-GST antibody, you can detect all the spots on this array where your protein is bound and determine sort of, what is the preferred motif? Where does this protein bind? And so this nicely matched, so this is just the wild type protein. This nicely matched the motif that we pulled out from all the ChIP-seq. And this also confirmed that that mutant protein does have a different preference for its motif. Now we turn to the RNA-seq data to, see what genes are potential targets, and are also being overexpressed in these tumors. And this is just looking at the plot of RNA-seq data here, looking at genes that are either overexpressed and under or downregulated within these tumors. And so this is looking at these mutant tumors versus the wild type tumors to see, again, this mutation specifically, what kind of programs is it driving? And one gene that really popped out to us, it was one of the few signaling pathways that was enriched in this data set was Wnt5a, so Wnt signaling, not specifically non-canonical Wnt signaling. And other players, I'm not going to talk about them, were also enriched within, but they have, some, they've been described to have roles in modulating non-canonical Wnt signaling. So we focused on Wnt5a because there was a lot of arrows pointing at it that this could actually be, an really important gene for this. And so the nice thing, Wnt5a receptors are actually expressed in the nephron progenitor cells. So the nephron progenitor cells, if they had this mutation, because they express the receptors, they would be receptive to an increased expression in Wnt5a and be able to respond to that signal. And this gene, Wnt5a, is actually not expressed normally within the human fetal kidney. So looking at single-cell RNA-seq data of the of the human fetal kidney, if you look down here at those nephron progenitor cells, right here, we see essentially no expression of Wnt5a. So this is a gene that's being specifically turned on in these cells that are carrying this mutation. And if we turn now to the ChIP-seq data, again, this is just from a single tumor, so the data quality isn't the greatest for this, but there did appear to be a peak near this Wnt5a in one of the introns, where there was an enrichment for both the wild type and the mutant protein. And if we take now this enhancer element and we put it into a luciferase assay, so just seeing if either the wild type or the mutant protein can actually, bind to this enhancer and turn on the expression of a reporter just in cell culture, it does show that both the wild type and mutant protein can bind to this enhancer. So there is potential that these proteins are driving then the expression of Wnt5a in these tumors. So as I mentioned, Wnt5a actually signals through the non-canonical cell pathway. So canonical being the one that has the transcriptional output and the transcriptional response where beta-catenin goes into the nucleus and turns on downstream genes, and non-canonical being the response that induces changes in cell polarity or cytoskeleton or migration or invasiveness. And Wnt5a, interestingly, has also been described in several different types of cancers to be involved in the oncogenic programs in these different cancers. And also the non-canonical signal can block the canonical pathway as well. And so it turns out canonical Wnt signaling is actually important for differentiating those nephron progenitor cells. So potentially, if there's some block to that normal differentiation, this could help be promoting that tumorigenic program. So this comes down to, can Wnt5a actually block the differentiation of those nephron progenitor cells and support their oncogenesis? And so we wanted to be able test some of these hypotheses now. And so these tumors are actually quite rare, and then getting the tumors with these mutations and being able to assess them, and if you actually need a homozygous tumor to be able to do any sort of ChIP-seq, to be able to differentiate the wild type and mutant proteins, there's a lot of issues with it. And so we thought, well, are there other ways that we can model this? And so the issue, like I said, current cell models, they're insufficient, they're highly transformed, and they actually don't represent the blastemal subtype that we're interested in. And then similarly, current Wilms tumor mouse models also represent the other subtypes. There is no blastemal predominant, model within a mouse model. And there was actually attempts, two different attempts to model blastemal predominant mutations in mice, but the groups found that they actually did not promote tumorigenesis. So even inducing these mutations in the mice, they weren't able to do that. And that could be because there's just different programs between mouse and human in those nephron progenitor cells that, so maybe we just can't model them in mice. So our alternative that we came up with is kidney organoids. And so, the nice thing about kidney organoids is they follow developmental programs and can be edited to carry disease-causing mutations. And it's been shown by other groups, specifically looking at cerebral organoids, they've been utilized to actually model pediatric cancers. And so, there was, the suggestion that this could actually be a nice system in which to model our Wilms tumor. And just to show you, so you can take iPS cells, so pluripotent stem cells, and push them down this pathway of differentiation and form little kidney organoids in a dish. And across this period of time, these cells will actually transition through a progenitor stage before those progenitors, nephron progenitor cells, differentiate and actually form these nephrons within this kidney organoid. So these are just immunofluorescence showing different markers of different segments of the nephron here. And as I said, we can take, our differentiation protocol, and the great thing about it is as you drive it through this differentiation program, they pass through this nephron progenitor stage. And so we can now look at gene programs modulated by transcription factors like 6-1 within these organoids and see now, what might be different between an organoid that harbors just wild type 6-1 or an organoid that harbors the 6-1 mutation associated with Wilms tumor. And so we engineered our iPSCs to have either the 6-1, just a wild type protein, or the Q177R mutation. And we also attached a flag tag to it, and also this iris that will allow independent translation of GFP. So now we can specifically use this handle, the flag tag, to isolate just the mutant protein, and then also use this GFP to be able to look at the cells or fact sort them in the future and do things. So this flag tag can actually enable now ChIP-seq, and the RNA-seq, the GFP label can allow us to isolate these cells and do RNA-seq. And we did this in a heterozygous fashion as well. So as I mentioned, the tumors are heterozygous, and it's very rare that they're homozygous. So we only introduced these cassette at one locus. So the other locus is actually wild type in either this mutant line or the wild type line. And so, if we take these cells, so this is just looking here at this mutant line we have, and we differentiate them to the nephron progenitor stage, we can see that they're expressing our 6-1 protein. They're also expressing that flag tag and the GFP. Additionally, when we try and isolate those proteins using that flag tag as our handle, and we're just blotting here, we can see that we can isolate either the wild type protein or that mutant protein from these organoids. And so now we can do the organoid ChIP-seq and look at, is this recapitulating these tumorigenic programs? And, or even just the normal developmental programs that you would see in these nephron progenitor cells. And so what we find, just looking here at, in this case, at the 6-2 locus, we know 6-1 binds. I showed you that previously, near this, near the promoter of 6-2, and there's also this very distal element as well. We can see the wild type protein is binding and the mutant protein is binding as well, suggesting that the wild type and mutant proteins are helping to drive a nephron progenitor program. And what was quite nice is when we compared our organoid ChIP-seq, the motifs that we recover from these ChIP-seq data sets, they match what we find either in the human fetal kidney or the tumor. So our organoid, 6-1 protein is binding to the same motif that it does in the tumor. And what we found, looking now at this, so here we're just looking at, for an example, now we've switched here to the 6-1 locus, where we know both the 6-1 wild type and mutant protein bind. And we look at this peak intensity here, we actually see that they bind about the same. So probably about the same binding affinity to this specific enhancer. But what we found at that previously identified Wnt5 enhancer in the tumor, so this is the one we looked at previously and actually tested in that luciferase assay, we find that there is binding of both the wild type and mutant protein, but the mutant protein actually seems to bind with higher affinity to that site. And we can actually observe that by ChIP qPCR as well. So we do this ChIP and we actually look for the relative enrichment of either the wild type protein or the mutant protein at that enhancer. And we see actually the mutant protein is more, you see greater binding at that specific enhancer, which actually may promote that, drive a high level expression of Wnt5a in these organoids. And going back to that motif and really trying to get at the mechanism, of what's happening with that motif, it turns out in that Wnt5 enhancer, there's this core sequence which looks very much like that mutant motif. So this is taken directly from that enhancer I just showed you back here. And the motif where 6-1 should be binding here, it actually looks more like the mutant motif with this GT-GT sequence here versus the wild type motif. And if we do an EMSA assay to look at the relative binding, so look at the binding of the protein to a DNA oligo containing that sequence, and do this gel shift assay, we see that the wild type protein does bind, but with pretty low affinity, whereas this mutant protein, you can see even at low concentrations, it binds to this motif. So this would actually suggest that, again, and why we see greater enrichment there, that it actually has a higher affinity for this site than the wild type protein. And if we now mutate that sequence and get rid of that preference for that G there, we can now normalize that binding and it becomes more like the wild type level, where you see about the same or equivalent binding of the wild type or mutant protein. And as I mentioned, this may be able to drive higher expression of Wnt5a. And it turns out that, that in these mutant organoids, there is actually greater expression of Wnt5a than the wild type. And so these are just some controls here, but really what I want to focus on is Wnt5a over here. So as we go across days of differentiation from day 11, 13, 15, when we compare the mutant to the wild type, at day 15, there's about a 6-fold increase of Wnt5a expression over what we see in our wild type organoids. And so, the question kind of comes down to, in our minds, do our organoids actually model Wilms tumor oncogenesis? So our hypothesis would be, if we're driving these tumorigenic programs within our organoids, and we differentiate them, we would expect that the wild type organoids would be able to go and form nephrons as normal in our little organoids in a dish. However, in the case of our mutants, our mutant line, we might predict that it cannot form nephrons, potentially due to that overexpression of Wnt5a and other factors, and it will actually maintain this progenitor-like state. And some preliminary data where we've actually done these differentiations now, looking now at the wild type organoid here that we've let go out and differentiate, we can see that aside from a few cells off to the side here, they differentiate as normal and form nephron structures within these organoids. However, in our mutants, we see that they don't really form any of these nice nephron structures, and they actually maintain these nephron progenitor-like cells that express 6-1, and again, that flag protein and GFP. And so now we're trying to really, characterize these cells and understand what they are, and, is this, recapitulating the tumorigenic program here. But we think we have some promising, preliminary data here that suggests that we can actually do this modeling with these organoids and get more insights into how these mutations drive tumorigenesis and come up with new therapeutic targets. And so just the model we have here, so we know that in the normal case, 6-1 and 6-2 help promote this maintenance of these nephron progenitor cells. And in the case of our P177R mutation, this is promoting the expression of genes such as Wnt5A, which then can lead to the continued self-renewal. And like I mentioned, non-canonical signaling can actually block the canonical pathway, which is normally required for this differentiation of nephron progenitor cells into nephrons. And so, is this the mechanism of what's happening? And I put Wnt11 here because Wnt11 actually is important for maintaining the nephron progenitors normally. Is Wnt5A sufficient, and can these cells ever be differentiated? So could we potentially, with these patients who have these blastomal predominant tumors that look like nephron progenitor cells, is there a potential that we could do a differentiation therapy with them rather than traditional chemotherapy? And perhaps that's something that could be an alternative strategy. And so for the last part of my talk, I just want to switch then to podocytes now, and a model now that we've developed to, be able to identify, potential therapeutic targets in podocytes that may help in this state of disease. So going from nephron progenitor cells now, we know that they differentiate down this trajectory where they form then these early epithelial structures that go on to pattern and differentiate into all the different cell types of the mature nephron. So this is just showing that they polarize, end up being segmented with proximal, medial, and distal segments here, and they form these different cell types of this nephron. And what I want to focus on is, in particular, these cells within this glomerular filter called podocytes. And so podocytes are really essential for kidney filtration. And so in here we have these blood vessels, these arterioles coming in, and the podocytes actually wrap around these arterioles within, or these capillaries within this glomerulus here, and are really a really important part of the filtration barrier here to keep all the essential things like red blood cells and albumin, circulating back into your bloodstream, but getting rid of the filtrates, and the things you want to eventually excrete in the urine. And podocytes are just this really beautiful cell in that sense that they have this cell body with these what are termed processes that come off, or foot processes, where you have primary and secondary processes that come off, and they form these really beautiful interdigitations with each other. And you can see this here, where it almost looks like they're kind of braided together, but they're really just interdigitating like this and helping to form this filtration barrier. And the way they do this is it's accomplished in part by the slit diaphragm structure that runs in between those interdigitating foot processes. So as I mentioned, they wrap around this endothelium, and the endothelium is actually fenestrated and has these little holes in it that will allow that filtrate to actually pass through this intervening glomerular basement membrane here, just made up of this extracellular matrix, and then through, this little molecular sieve here. And so there's these protein bridges, as I mentioned, they were called slit diaphragms, that help to regulate, the filtration and help to, keep things in the blood that you want to keep in, but get rid of that filtrate and move it down into the rest of the tubule. And there's a lot of proteins that are involved in this very, complex and beautiful structure that help form these foot processes and help to maintain and build these slit diaphragms. And I'm not going to go through all of them, but just to show you that there is, a number of proteins we know that help to, stabilize and promote and keep those slit diaphragms together. And these are very actin-rich processes. So actin is really what's helping to really build the structure and maintain those foot processes. And this is just looking now, if you looked kind of in between, and these are those slit diaphragm proteins here that are really important, again, for maintaining that, that filtration barrier, but also it helps to maintain the podocyte architecture and keep them interdigitated. And so we do know a lot of players and how they link to the actin cytoskeleton and how, a lot of this is modulated. Oh, I should show here again, just looking on EM, because I'm going to show an EM picture in just a few minutes. If you take a section through this, you can see here is a podocyte, and these are all these little foot processes here lying over. So this is a capillary lumen here, and lying over this glomerular basement membrane. You can see this here in this zoomed in view. So these are actually the foot processes of these podocytes, and they form this sort of beautiful beads on a string, when you see them in EM. And you can actually see those protein slit diaphragms within these EM images. And again, this is just those fenestrated endothelium here. You can see there's actually holes in the endothelium where now the solutes will pass through up into this urinary space here. And so, I said that podocyte architecture is really critical for helping to maintain, that filtration barrier. And it turns out that in many kidney diseases, irregardless of the cause, that there's compromise to that filtration barrier, which eventually, leads to a progression in the disease, and also the proteinuria that we often test for in the case of kidney disease. And so, this again, looking at this normal case here, we see these beautiful interdigitations of these foot processes, and you can see in the schematic here. But what happens in the case of disease is that these podocyte foot processes actually undergo this effacement, and they flatten out, and they lose those nice interdigitations. Those slit diaphragms are now compromised, and you're going to now end up losing that really nice filtration barrier, and you're going to get protein leaking out into that urinary space. And this is what will end up coming out in the urine, and what we test for is looking for that protein in the urine, and that would again suggest that there's compromise to that filtration barrier. And just this is nicely schematized here. So again, those proteins would normally not make their way through these endothelium, through these podocyte foot processes and their slit diaphragms. But in the case of disease, the podocytes efface. Eventually, they'll undergo apoptosis or detachment, and now you're going to get those proteins coming out into that urinary space and out into the urine. And so, can we come up, with therapeutic targets where we could actually, potentially prevent the progression and that effacement and loss of the slit diaphragm structure, or potentially treat, people with this disease? Can we prevent the loss or even restore that very unique and important podocyte architecture in the case of disease? So an ideal target would be something that's a component of the slit diaphragm or involved in maintaining slit foot process integrity. And so our goal was to identify the spatially localized proteome of the slit diaphragm in both health and disease to identify possible targets. So, certainly there's a whole bunch of RNA-seq data out there that has, compared, normal, healthy, and disease states. And we've gained a lot of insights from there, but the RNA-seq data doesn't tell you about where these proteins localize within any cell, right? So how do we know those, how of those proteins, those genes and proteins are actually important for maintaining the podocyte foot process integrity or whatever other process you're looking or interested in? So we turn to proteomics then as a better way to address this and find important players right there in the space we want. And how can you do that? So if you know anything about trying to do proteomics and isolate specifically membrane proteins too, it can be extremely difficult to isolate these, extract them from the membrane. It can actually, because you're going through these harsh conditions, you'll lose a lot of those interacting proteins. And so you won't, it can be very difficult to get the full proteome of what's happening. And so recently, within the last decade, a lot of these proximity dependent proteomic approaches have popped up. And specifically in this case, this BioID approach, is able to identify spatially localized proteomes. So what's great about this is that you can attach this promiscuous biotin ligase as a fusion to your protein of interest, and then identify all the proteins that interact. And this was actually originally developed by Kyle Rue's lab at Sanford Research and University of South Dakota. And it's been used many times over now, and most specifically in cell culture. It's very easy to do in cell culture to be able to transfect in your fusion proteins and add in some biotin and get this to work. And recently, a lot of groups have started to use this to map out, the whole interactomes of cells, and even, looks very specifically at, more specific localization, let's say in this case of synapses, and looking at the complexes that are involved in synaptic processes. And this was the first paper really that had done it in vivo. And really to this day, a lot of studies, I'd say, 99% of studies are still done in cell culture. But we wanted to make a mouse model, and we had no idea if it would work, but we thought, well, let's take a chance and see what happens. And just going back then to this, how does this work? How does this BioID protein work? So you have your protein of interest here, so in yellow. So let's say, this is your protein. You want to find the proximity of anything around it, the localized proteome. You fuse it to this BurA, which is this bacterial biotin ligase, and it's been engineered so it's promiscuous. And so essentially anything it comes into contact with is going to slap a biotin molecule on it. And the fun thing about this is you put this little linker in between your protein and the BurA enzyme, and you can actually have this flexible space. In this case, it's 10 nanometers, but you can get up to 25 nanometers of a radius from your protein of interest to really, get an idea of what that localized proteome is. So you're going to tag proteins with biotin that are in direct contact with your protein, and you'll even tag your own protein. But you'll also get proteins that are within the radius. They may not have a direct interaction with your protein, but they do come within the proximity of your protein at some point in time. And so this biotin, is stable, and it's going to stay on that protein for as long as that protein is still stable within those cells. And so you can take this and lyse the cells and do a really harsh extraction. It does not disrupt any of the biotinylation on these proteins. So you can extract everything, membrane proteins, everything from here, and then purify out those proteins with streptavidin. And so now you have a pool of isolated biotinylated proteins that you subject to mass spectrometry, and you can get the proteome. And so, as I mentioned, we had no idea if this would actually work in vivo, but we took the chance of making this mouse. So we used podocin as our protein that was localizing to that slit diaphragm. And we attached this BurA biotin ligase to it. There's also an HA tag there, which is really nice. It makes a great marker and handle on this protein. And we use this 13X linker, which actually gives about a 25 nanometer radius from podocin, where it's going to tag anything with biotin. And so we made this mouse. We introduced this to the actual podocin locus. And so the idea would be that you have podocin sitting here. Podocin actually is not, that external part of that slit diaphragm. It sits right here internally. So it's going to tag anything internal in that podocyte foot process that's in the local vicinity of that slit diaphragm complex. And it's going to tag those with biotin, and we can isolate those biotinylated proteins and subject them to mass spec. Oh, first off, we had to verify that this was actually working, that it's producing our protein and it's localizing properly. And so using that HA tag that's on that, the BRA enzyme here, we can localize that enzyme and see that it's overlapped with podocin. So it's localizing, being expressed by those podocytes. And if we look here in EM images now where those slit diaphragms should be, so this is using an anti-HA antibody to immunolabel here, immunogold labeling, and you can see that that fusion protein is localizing to those slit diaphragms. So now we have verification that it's localizing, it's being made, it's localizing properly, which is great. But the question is it active? Can it biotinylate proteins in the proximity? So we inject these mice with biotin to induce that biotinylation of the proximal proteins. And so we inject either a wild type or our bioID animal, and we can see this is just verifying again the HA expression in our bioID animal, looking at podocytes here marked by WT1. And we can see when we label them with streptavidin, which is going to detect those biotinylated proteins, we see a significant enrichment for biotinylated proteins within those podocytes. And if we isolate those biotinylated proteins and run them on a Western blot, we can see that there's much more biotinylated proteins coming out of kidneys that were isolated from our engineered animals versus our just wild type controls. And if we probe now for proteins that we know should be within that biotinylated, so podocin with that tag, it should actually be able to biotinylate itself, and podocin also dimerases, so we would expect that we might see the other, unmodified podocin protein come up in here too, and those served as really nice controls that when we blot for HA in those biotinylated proteins, we do see the podocin bioID protein there. And then if we look and just probe for podocin now, we see the podocin bioID protein, but we also see podocin itself coming out. So, within these isolated proteins, we're identifying components that we think should be there. So we took and did mass spec to look at, what are all the proteins that are sitting there. And, we know some that should be there. And so we have some nice controls built into this, and we found certainly proteins that we know are there. So proteins such as this chiral, tight junction protein 1, again, podocin came out, that's great. And many of these genes have already been described to have roles and be important for podocytes and podocyte integrity and maintaining that filtration barrier. And this is just mapping these out and looking at their relative enrichment. And so, we did find a lot of, candidates here for things to follow up on. And ideally now we'll be able to compare this to disease models and look and see if there's differences in that localized proteome and, how that might change with disease. And we can also do this. We could look at the phosphoproteome or glycosylated proteins to really see how those events may be changing as well with disease. And if we look at just gene ontology enrichment, we definitely enrich for things that are part of, cell junctions or adherence junctions or involved in sort of cytoskeletal organization, which makes sense because those slit diaphragms help connect to that actin cytoskeleton and maintain that foot process integrity. So this really helped us validate this data set that it was really enriching for the proteins we're interested in. But, we also wondered if there was anything novel in here. And it turns out that we did identify a couple candidates, and I'm just going to focus on this one and just a couple for these slides here, just to show you that we can identify novel components of this too, that we don't know everything about the slit diaphragm quite yet. There are other players there. In this case, ILDR2 or immunoglobulin lactamine containing receptor 2 came out in this, and there's not much known about ILDR2. It's been most well described as being actually a negative regulator for T cells and having immunomodulatory roles, which is quite interesting. We're wondering if it might have some role in podocytes doing that as well. But it's also a component of tricellular tight junctions. So it's known to localize to tight junctions. So that kind of makes sense in terms of the podocytes and those junctional slit diaphragm complexes. And very nicely, just to confirm, it is expressed turning to single cell RNA-seq data. We see it is both in human and mouse podocytes, and very specific to the podocytes compared to all the different cell types in the kidney. And as I mentioned, it localizes to tricellular junctions, and this was shown in cell culture. So tricellular junctions being the points where 3 different cells come together and at this junction. So it seemed to be important for maintaining that. And interestingly, it may be important not at all spots in the, not at all slit diaphragms, or at every point in the podocyte foot process, because we actually see a punctate localization of ILDR2 here. So ILDR2 is in magenta, and we see it, localizing with podocin, but not complete, everywhere podocin is. So again, is it at very specialized junctions in these podocytes helping to maintain that architecture? And so we're actually currently assessing knockouts of these animals to assess the role of it. And for the last slide here, I just want to touch on, as a, we haven't done our disease model yet, but are there things we can look at in aging? So we had these mice kind of hanging around, as we were doing all this bio-ID, we kept a few around. And by the time we, got done with characterizing the mice and making sure that they were working, we had some that were about 2 years in age. So we thought we might as well do the proteomics on those and compare those to our 6-week-old mice. And just to show a few proteins that popped out of this, and we show that there are some proteins that are becoming more enriched at the slit diaphragm in these aged mice. And there's also proteins that seem to be going down in their enrichment at that. And interestingly, the ones that seem to be coming up are increased in tight junction associated proteins and even desmosome associated proteins. So, this might be an attempt at these podocytes as they're aging to keep even tighter, them tighter, more tightly together, so that they can maintain that filtration barrier, because this actually gets compromised in aging. So maybe this is their attempt to build these strong, tight junctions. And tensin is actually important for maintaining podocyte attachment to the glomerular basement membrane. So this may suggest that there's also this sort of looseness coming here. And I just want to show you that tight junctions are normally involved very early on in podocytes. They kind of keep them together before they develop and flatten out and get those really nice, beautiful foot processes. So they're, an increase in tight junction proteins could suggest that they're more, something more like this early developmental phenotype here in an attempt to keep these cells together. But with that, I will end, and, hopefully we'll have some more data on all these disease models to share the next time, but definitely just want to, it's really these 2 graduate student and postdoc who have done all the work. So Matt Stevenson did all the organoid work, and Gary Gerlach did all the podocyte bio-ID work. And they're really, the 2 that drove each of those projects. So thankful for them for, generating these models and making this beautiful data, and then all our collaborators and funding sources. And with that, I'll take any questions.
That was great. Thank you so much. That was so interesting. So if we visit your lab, do you take us to go throw axes? Will that happen?
Absolutely.
Good.
Yes.
So there's a question from Charlie Alpers, and he wanted to know if you found nephron at the slit diaphragm in your bio-ID mice.
We did not, interestingly. We thought, we might, and I don't remember specifically how big that intracellular domain for nephron is, or, so there may be some limitation there on podocin to be able to actually interact, but we did not find it in our data. So we're a bit surprised, but maybe there's something with the conformation and everything there and localization that we just couldn't actually detect it.
That's interesting. Erica has a question. Go ahead, Erica.
Yeah, thanks. That was really nice, really beautiful. I had a question. I was intrigued by this concept that the nephrons are developed by 36 weeks in utero. So premature babies must have very undeveloped kidneys. And I was wondering what kind of treatment they would undergo and how this informs sort of your studies. How do you make these nephrons differentiate? How do you treat this sort of scenario?
Yeah, I mean, that's a fantastic question that so babies born earlier than that, there is some evidence that they may undergo premature cessation of nephrogenesis. So once they're born, they may actually deplete those nephron progenitor cells faster than if they had stayed in utero. And they'll end up potentially with fewer nephrons overall than counterparts that go to normal gestational age. Additionally, there's been studies in baboons and stuff that have looked at this too, where they looked at early birth and they see that the glomeruli, which are doing all the filtration, actually look bigger, and there's hypertrophy of them. They maybe because they're working harder, maybe a consequence of some of that early birth. But there really is nothing to do about that. I mean, it doesn't seem to affect, kidney health at that stage. Later on, as they get older, having fewer nephrons could set them up as being more predisposed to hypertension or kidney disease. But yeah, at that point, they'll still be fine. The kidneys they're going to work fine, and they're going to filter fine, even with this early cessation and reduced number. But it's a great question that many people are interested in. Is there a way we could actually extend that period in these premature infants so that they could have a longer period to form more nephrons? And we have no idea how to do that yet. But definitely an active area of investigation.
So I have a quick question about the bioID mouse. So what is the sort of labeling time that you have for injecting the biotin? So
we inject them over, I didn't show those details, but we inject them over a 7-day period. It's kind of what we found in the literature from about the 2 examples that ever tried it in vivo. And so they did it 7 consecutive days, 1 injection each day, and that's sufficient to get enough biotinylation. But you could definitely change that window to do shorter time periods, catch, more transient interactions, or keep it going out for, a long period, do it multiple times. So it can be modulated in that way too. And yeah, you just choose whatever time point you want to do the injections, which is nice. So it can be temporally controlled.
Have you seen any disruption of the foot process at all after the injection?
We did not do EM after the injection to see if there was anything. So we don't know. It's a good question. Something we should probably just ensure, that everything is still maintained. But we haven't observed anything that looks different after looking at, just on sections. Glomeruli seem normal, but we should probably do that EM just to make sure.
Yeah. And one more question from Charlie, and I think we'll probably be out of time.
So my question is, it's in adults, it's virtually unheard of to find tumors of podocytes or parietal epithelial cells or differentiated glomerular cells. And do you relate that to the suppressed expression of 6-1 or 6-2 or its signaling pathways? Or do you have other ideas about that?
Yeah, that's really interesting. I hadn't really thought about that, in terms of that downstream nephron progenitors and giving rise to those cells, and whether that's has to do with suppressed programs. That's a really interesting point. I don't know. It's certainly a good hypothesis that it could be with that, because yes, you don't see any 6-1 or 6-2 expression normally with an adult kidney. And so anything downstream of that, you might not expect to see downstream consequences to other cell types, differentiated cell types from those nephron progenitor cells. But yeah, that's an interesting question. And yeah, one I don't have the answer to, but interesting hypothesis there. Yeah.
All right, very last question from Ram. He wants to know, is Wnt5a upregulated in other tumor types, and is it sufficient for oncogenesis?
Yes, it is upregulated in other tumor types. I'm trying to think of exactly what ones right now. kind of blanking on that, but it has been found in non-kidney tumors, not just kidney, but in other, cell types. And I think, the question, is it sufficient? I don't know if they've tested that so much in some of these models, but they have tried to treat them with inhibitors of Wnt signaling, and it is sufficient in some cases to, cause those cells, tumors to shrink or things like that, or cause reduced proliferation. So there is potential that it could be, one of the primary drivers of that program in other tumors, but I don't know. And it's still a great question whether it is in ours as well. Is this solely responsible, or are there other important players?
All right. Well, thank you so much. That was great. I'm going to email you about cofillin in old mouse slit diaphragms, because I've found that as well with some proteomics I've been doing with glomeruli. So
Interesting. Very interesting. Great.
Yeah, I'm really curious to share data. But anyway, it was such a pleasure to have you here, and I hope next time we can get you out in person.
Yes, that would be wonderful. I'd love to visit. And thank you so much again for everybody for attending and the great questions, and really appreciate the opportunity to share our work.
Right,
Yeah, thank you for giving that great talk.
Great.
Thank you, Lori.
You're welcome.
you.
Thank you.
Bye.